Hello, everyone. My name is Tyler Armstrong. I'm a graduate student in Dr. Lund's lab, and I'm very excited to present this today. I'm presenting on Alzheimer's disease, the progression and treatment of dementia. Dr. Lund's lab studies air pollution, and one of our new focuses is going to be air pollution and its effects on Alzheimer's disease. So this was a great way for me to kind of brush up on the topics that I need to know for my research. So let's get started here. Alzheimer's disease is the most common form of dementia and is the fourth leading cause of death in adults over the age of 65. The main pathologies of Alzheimer's disease are the presence of amyloid beta plaques and tau neurofibrillary tangles accumulating in the frontal cortex region of the brain, resulting in atrophy of white matter area. These pathologies result in clinical symptoms of dementia, such as cognitive disorders, loss of memory, loss of personality, physiospatial disorientation, and difficulty swallowing. A patient may not necessarily die because of Alzheimer's disease, but because of complications with the disease. For example, as the disease progresses, patients have difficulty swallowing. They can improperly swallow, resulting in food being deposited in the lungs, resulting in infection, or choking on food because of inability to swallow. Alzheimer's disease was originally named after the German psychiatrist Dr. Alois Alzheimer, pictured here to the far right, who in 1906 treated a patient by the name of Auguste Dieter, shown on the left. She was the first person to officially be diagnosed with Alzheimer's disease, even if it wasn't called that at the time of the diagnosis. She was born in 1850, and she started showing signs of Alzheimer's in her early 50s. She had trouble with her memory, she couldn't sleep, and was having problems with language and writing. These were all signs of dementia, but she was very young to be presenting these symptoms. She was brought to Dr. Alzheimer by her husband after she accused him of being unfaithful to their marriage and creating delusions in her head about an affair that he might have been having. Due to the stress of the situation, her husband brought her to the Institution for Mentally Ill and Epileptics in Frankfurt, Germany, where she was under the care of Dr. Alois Alzheimer. Dr. Alzheimer initially diagnosed her with presenile dementia, but was fascinated by her disease because of how young she was and the severity of her symptoms. She could answer questions about what her name was, but if she asked what her husband's name was or one of her friends, she would simply reply, Auguste. No matter what she was eating, whether it was pork, soup, or pasta, everything she ate, she always said was spinach. She could recall the names of objects shown to her, but she could not recall the object after it was out of her sight. So you could hold up a pencil to her and she'd tell you, that's a pencil. But if you took the pencil away, she wasn't able to tell you the last object that you had shown her. Some days she was more herself than others. And when asked how she was on a good day, she would say, I've lost myself, so to say. That's one of the quotes that she's documented saying when she was in treatment. She stayed at the institution for 5 years until her condition worsened to the point that she had lost most of her cognitive functions. And then on April 8th, in 1906, she died. Dr. Alzheimer ordered an autopsy after her death, and her brain revealed an accumulation of plaques and tangles of fibers, as well as atrophy of the frontal cortex. These subsequently became known as amyloid beta plaques and tau neurofibrillary tangles, which are still used as biomarkers in the treatment of Alzheimer's today. So that was about a century ago. So we've had a century of progress so far on Alzheimer's disease. In 1911, doctors across Europe and the US started using Alzheimer's diagnosis criteria. Dr. Emily Redlich names the disease after Dr. Alzheimer in his 8th edition of Psychiatry, but it's probably pronounced a different way because of the fancy italices. In 1968, American Psychiatrist Association's Diagnostic and Statistical Manual of Mental Disorders concluded that the degree of neurofibrillary tangles was correlated with performance on standardized cognitive tests, which led to the use of those standardized cognitive tests in the diagnosis of Alzheimer's disease. Around 1984 is when the National Alzheimer's Disease Research Center was established. through funding from the National Institute for Aging. In the 1990s area, that's when we started to expand the diagnostic criteria of psychological characteristics of Alzheimer's disease. And then in the 2000s, right when we're getting started with the Human Genome Project, is when we started to really uncover all the genes that are correlated with Alzheimer's disease as we gained more knowledge about the human genome. In 2010, in more recent years, this is the time when biomarkers have become a big thing. And this is the same thing for Alzheimer's. There are a lot of biomarkers that became popular in areas of research and diagnostic at this time. Diagnostic techniques have greatly improved since Dr. Alzheimer's original diagnosis. Now, in conjunction with psychological tests to determine whether or not the patient is suffering from dementia or just mild cognitive impairment, there are also biomarker methods such as measuring the presence of amyloid beta plaques or tau fibers in the cerebrospinal fluid. Neuroimaging can also be used to determine whether or not the patient is presenting physical signs of dementia and gives insight into how far along they are. It could be pre-dementia or mild cognitive impairment, which is usually the beginning stage of Alzheimer's disease. After that, Alzheimer's disease dementia can be classified as either early, middle, or late. To the right here, we have a selection of MRI scans taken from patients with varying degrees of Alzheimer's pathology. They are overlaid with voxel-based morphometry, which is fancy speak for the computer figured out the changes in density. So the red is the bad part. As you can see, the patient at the top with mild cognitive impairment is beginning to show decreases in brain matter, and as you progress to the late stage on the bottom row, the brain has atrophied significantly. Diagnostic techniques like this are great for determining how far along the disease is, and allows us to physically observe changes that in Dr. Alzheimer's time could only be done post-mortem in an autopsy. There are also neuropsychological tests that can be used when the diagnosis is unclear, such as the National Institute of Neurological and Communicative Disorders in Stroke and the Alzheimer's Disease and Related Disorders Association criteria. On a side note, someone should contact a PR representative there and talk to them about more compact acronyms. They group Alzheimer's diagnoses into 4 categories. The first is unlikely Alzheimer's disease, when the patient has suddenly presented signs of dementia, is having seizures, and has a sudden onset of neurologic signs, but might not necessarily be Alzheimer's disease. The second is possible Alzheimer's, when the patient has syndromes of dementia with slightly atypical onset, but no known cause, and they don't have any other morbid diseases that are capable of producing dementia-like symptoms. The third is probable Alzheimer's disease, where dementia has been established. They've passed the first 2 criteria of unlikely or possible, and there are very clear signs of cognitive impairment in 2 or more areas of cognition. The patients usually have to be between the ages of 40 and 90, and it has to be confirmed that they don't really have any other diseases capable of producing these symptoms. The fourth criteria is probable Alzheimer's disease. And to get to this level of diagnosis, you usually need an autopsy or a biopsy that's confirmed histopathological evidence of Alzheimer's disease, such as the presence of amyloid beta plaques or tau neurofibrillary tangles. So Alzheimer's can roughly be categorized into 4 categories based on the cause of the disease, although there's significant overlap in these. There's late onset Alzheimer's, abbreviated as LOAD, early onset, familial, and sporadic. The cause of late Alzheimer's disease and early onset Alzheimer's disease are not fully understood, but there's strong evidence that people with one isoform of the cholesterol packaging apolipoprotein E, abbreviated as ApoE, are more susceptible than others. You could have ApoE 2, 3, or 4. People with the ApoE4 allele represent 80% of late onset Alzheimer's, and 40 to 50% of early onset Alzheimer's. Familial is a type of early onset Alzheimer's that is usually associated with inheritable mutations in the gene for the amyloid precursor protein, abbreviated APP, and also the presenilin genes, PSIN1 and PSIN2. Having one or more of these mutations greatly increases your chance of developing Alzheimer's disease later in life. Sporadic onset Alzheimer's disease is not fully understood, but it's thought to be a mixture of genetic polymorphisms such as ApoE. But there's also hypotheses that there could be viral factors that lead to the transmission of prions capable of inducing Alzheimer's-like pathology. There might be metabolic factors due to other comorbidities such as diabetes. Some scientists actually refer to Alzheimer's disease as type 3 diabetes because of the similar pathologies. There's also environmental factors that come into play, which is also where I come into this equation, since that's what I research in Dr. Lund's lab. The theories behind Alzheimer's disease, they're still under investigation and haven't led to any optimal treatments that target a lot of the mechanisms that induce the disease. As of right now, the only treatments available on the market are those that improve symptoms of impaired cognition without actually halting the progress of the disease. The two main theories behind Alzheimer's are the amyloid cascade hypothesis and the tau hypophosphorylation hypothesis. So here we have the amyloid cascade. The amyloid cascade hypothesis was the first hypothesis for the pathogenesis of Alzheimer's disease and is based on the presence of the amyloid beta plaques in the brain after autopsies. In this pathway, the amyloid precursor protein, APP, is cleaved by an enzyme to form a transmembrane amyloid beta. And this can be accomplished in 3 different ways that result in either normal physiology or Alzheimer's disease pathology. So the first one here is the non-amyloidogenic pathway, which produces neuroprotective elements, such as this C-terminal fragment alpha. These get embedded in the plasma membrane and can be neuroprotective. The second is the amyloidogenic pathway, where APP is cleaved to CTF beta by beta secretase. This results in various links of the amyloid beta being cleaved by the gamma secretase here and released into the extracellular environment. Once in the extracellular environment, they can aggregate together and accumulate around neurons, resulting in Alzheimer's disease pathology. The third pathway is the ATase secretase pathway. That is the alternative to the alternative normal physiological outcome that doesn't result in the accumulation of amyloid beta plaques. So there have been several drugs that have been proposed to target this pathway in an attempt to reduce the amyloid beta formation. For a while, beta amyloid converting enzymes and gamma secretase inhibitors were pretty popular for drug developers. Unfortunately, none of these drugs to inhibit either of these targets ever passed FDA clinical trials. This is usually due to a lack of effectiveness. Some of them had a plethora of adverse side effects and vital organ toxicity due to lack of specificity in the drug for neuronal targets. There are still new agents being proposed for this mechanism, some of which are currently in phase 3 clinical trials. The second hypothesis is the tau phosphorylation hypothesis. So tau is a microtubule-associated protein that's found in neuronal axons in the brain. Its normal physiological function is to maintain the microtubule structure and maintain the microtubule structure and function. Normally, the tau protein is not highly phosphorylated, but under pathological conditions, it becomes hyperphosphorylated. When it's hyperphosphorylated, you get these changes in structure. They start to get all wound up on each other. And as a result, you now have defective microtubules since they're no longer supported by the properly configured tau. As these defective microtubules and tau accumulate, they start to form the neurofibrillary tangles, and these get deposited uh, on the neurons. This is depicted here on the right side. As these neurofibrillary tangles get deposited on neurons, they reduce the number of synapses. They can cause neurotoxicity and result in overall cellular dysfunction. There have been drugs to target this pathway in a similar way to targeting the amyloidogenic pathways, such as targeting and inhibiting key kinases involved in tau aggregation, or to attempts to stabilize microtubules. Unfortunately, a lot of these drugs have met the same fate as the ones for the amyloidogenic pathway and did not actually pass FDA clinical trials. Because the mechanisms of pathogenesis are not fully understood, many of the proposed drugs have failed clinical trials, even though it's long been known that amyloid beta and tau are some of the main pathologies seen in Alzheimer's disease. There are still many factors here that aren't fully understood and are being investigated by researchers in academic, uh, pharmaceutical, and private industries all across the world. The causative agent of Alzheimer's disease is fairly undefined at this point. There are several known comorbidities and risk factors for Alzheimer's disease, such as diabetes, hypertension, smoking, obesity, dyslipidemia, just to name a few. Cerebrovascular disease has been shown to be one of the top risk factors for Alzheimer's disease. People that suffer strokes are, especially ischemic strokes, are at a much higher have a much higher chance of developing Alzheimer's disease following their stroke. So there's also increasing evidence that there's an environmental aspect to the progression of Alzheimer's disease, specifically sudden onset Alzheimer's disease, where they are lacking some of the usual genetic features, such as ApoE in the patients that develop Alzheimer's disease. This is actually my area of research at UNT. I work in Dr. Lund's lab, where we examine the mechanistic effects of air pollution. We use cell culture and mouse models with young and aged mice as a translational approach to looking at the effects of air pollution and how these actually end up in pathologies related to Alzheimer's disease. So I can take a section of a mouse brain and use fluorescent antibodies to target like the beta amyloid plaques and look and see where they're getting localized at inside the brain tissue. I was planning on showing some of my preliminary data to you guys in class, but I value my life and fear Dr. Lund too much to put unpublished data on the interwebs for everyone to see. So if you are interested in knowing about any of that, just reach out to me and I can show you. One air pollution researcher, Dr. Lilian Calderon Garcedunas, whose names I've just entirely butchered. She's a leading expert in this area. She's an MD and associate professor at the University of Montana. She studies the effects of air pollution on the progression of Alzheimer's disease. She has numerous publications about her autopsy analysis in Mexico City. The autopsies revealed that both children and adults had increased levels of amyloid beta plaques, early signs of neurodegeneration and neuroinflammation. And for those of you that don't know, Mexico City is one of the most polluted areas on the planet. The particulate matter count in the air is ridiculously high. And having that high count of particulate matter in the air has been linked to a lot of other diseases. Researchers like Dr. Lillian and Dr. Lund have been researching this for a while and have linked air pollution to other diseases such as ischemic strokes, hypertension, and cerebrovascular disease. Recently, Dr. Lund published papers on alterations in the blood-brain barrier as a result to inhaled traffic-generated air pollution. If anyone gets a chance to check out my references, there's some pretty interesting reads, or contact me and we can chat about it. All right, so now that everyone's anxiously holding their breath, let's talk about some of the drugs. So, current FDA-approved treatments are almost exclusively comprised of cholinesterase inhibitors, such as donepezil, galantamine, and rivastigmine, as well as N-methyl D-aspartate, abbreviated NMDA receptor antagonists, such as memantine. These medications do not treat the underlying disease, but may relieve some of the symptoms and slow the progression of Alzheimer's disease. There are 5 commercially available drugs that you can go pick up at the pharmacy. So 3 of these are donepezil, rivastigmine, and galantamine for the cholinesterase inhibitors. And then we have memantine that is our NMDA receptor antagonist. Tacrine was discontinued back in 2013, right when I started working in pharmacy. I remember having to go pull it from the shelves and dispose of it. So that's real interesting. That you also have one combination product. It's Namzaric. It's a combination of donepezil and memantine. It interestingly coincided with memantine becoming available as a generic instead of just the brand name Namenda. It's a pretty expensive drug, but you can probably just get it prescribed as 2 separate ingredients at a much cheaper price. So let's talk about cholinesterase inhibitors. Many researchers have found that there's impaired synthesis and secretion of acetylcholine in the cholinergic neurons in the cerebral cortex of Alzheimer's disease patients. So this can result in impaired memory and cognition, as they're not able to fully transmit signals between neurons. Cholinesterase inhibitors are used to increase this activity through the non-competitive inhibition of acetylcholinesterase. This results in better management of the little acetylcholine that is being secreted because it's not broken down as quickly into choline and acetate. So there's more opportunities for the acetylcholine to interact with its postsynaptic nerve terminal. The design of these drugs is based on the mechanism of acetylcholinesterase binding to acetylcholine by targeting the serine hydroxyl group at the esteric site and having a tertiary amine at the catalytic anion site. Notice how most of these drugs here, galantamine and rivastigmine, have -ine at the end. That's because of this tertiary amine that is present on most of these drugs. Unfortunately, this drug cannot cross the blood-brain barrier in its protonated form. They can only cross over in the neutral form readily. But in order for them to interact at this catalytic ion site, they need to be in the protonated form in order to actually make binding interactions here. So this can result in some efficacy issues if some of the drug is not able to get into the central nervous system where it exerts its effects. All right, and then we also have memantine. So memantine, like I said earlier, is an NMDAR receptor antagonist. So these work primarily through glutamate. So overstimulation of these NMDA receptors by glutamate has been found to be very common in neurodegenerative diseases such as Alzheimer's disease. The large amount of glutamate binding to the receptors opens up channels that allow for an influx of calcium into the neurons. When you have too much glutamate, you now have too much calcium entering the neurons. An excess amount of calcium can be cytotoxic and produce free radicals that result in neuronal damage. This clever picture here put radical humans protesting something to represent these free radicals. So that's what you see depicted here. But after this channel is open, memantine can block this to prevent further damage to neurons in the central nervous system that are already being degraded by Alzheimer's pathology. So these NMDARs are ligand-gated ion channels that have 4 subunits. They contain one NR1 subunit and an NR2 subunit. In order for the channel to function properly, glutamate residues and of the NR2 subunit and the glycine residue of the NR1 subunit must be occupied in order for the channel to open and allow the influx of calcium. After the channel has been opened by the ligand binding at NR2 and NR1, memantine can enter the channel and bind at this M2 region of the NR1 and NR2 subunits. Because it only does so after the channel has opened and it is binding to an enzyme substrate complex, this can be classified as an uncompetitive antagonist, which, as we said in class, is a pretty rare species of antagonists. So pretty interesting. As of 2019, there are 132 agents in clinical trials for Alzheimer's disease that are beyond the scope of these symptom treating cholinesterase inhibitors and NMDARs. 30 of these are in phase 1, 74 are in phase 2, and 28 are in phase 3. Of the 28 in phase 3, 3 are symptomatic cognitive enhancers, 8 target neuropsychiatric symptoms, and 17 of these are disease-modifying therapies. Many of these new targets actually have anti-amyloid properties to them or anti-tau properties, trying to target the mechanisms that we discussed earlier. They haven't passed approval, but they are in the final phase of it. Some of the drugs in some of these other areas, such as neuroprotection, anti-inflammation, or antibiotic, sleep disorders, those are actually targeting, or those are drugs that have already been established for the most part. Some of them are indications, or some of them are disease-modifying therapies, such as the blood pressure cholesterol combination drug, losartan, amlodipine, atorvastatin. What they're trying to do here is target the underlying cerebrovascular disease that I was discussing previously. By treating the disease preemptively, they're hoping that they can modify or at least slow down the progression of Alzheimer's disease using already approved drugs, but these clinical trials are trying to expand the indications for which these drugs can be prescribed. So if anyone has any questions or comments, feel free to message me on the discussion board or shoot me an email. I'd love to talk to you guys about this if you have any questions. Thank you so much for your time. And here's my references. All right.